So let me also thank, of course, there's Dr. Pradeep, and we all extend our thanks to him for spending so much time to share his decades-long experience in vector surveillance, vector control, all the strategic planning he's done. It's not just for malaria, but he's done it for many other aspects as well, for lymphatic filariasis, etc. So he's got an excellent idea of the big picture and what one needs to consider and how they interrelate and how you need to bring them together to adequately, effectively achieve the best vector control that you can with your specific resources available. So thank you, Dr. Pradeep. I'd also like to thank our team in the background that makes sure everything's running smoothly. There's Dr. Pon in Singapore from APMEN. In Bangkok, we have Aldila Sabiha Wulandari and also Dr. Popo Aung. Thank you to the two of you for your support. I'd like to mention that as you've noticed, the event is being recorded, and that is, the preceding 2 days have already been uploaded. I think Wulan, if you don't mind, just pop the link into the chat box so that our viewers may want to inform friends or other people that may not have been able to listen in, so that those links can be shared with them. So the events are being recorded. They are available also for our audience. Please, as Dr. Pradeep proceeds with his teaching, if you have questions that come to mind, pop them into the Q&A box, not the chat box, please. We'll be monitoring the Q&A box, so slot them in there as soon as they come to mind, because then you're high up in the ranking. Because we often can't finish all the questions, so at least we get to the first half or so of the questions that are posed. Just a quick word about process. So again, it will probably be about 1 hour of presentation, followed by a 10-minute comfort break, another hour of presentation, roughly, give or take, and then about a 20-minute or so Q&A session where we run through the questions in the Q&A box. This is day 3 of our excellent 4-day series of presentations on strategic planning for vector surveillance and control, given to us by an expert in the field. We're midway through it now. This is day 3. Tomorrow is our last day. And again, thank you, everyone. Over to you, Dr. Pradeep.
Thank you, Leo. Let me share before we start for today's, just for connectivity, as we have done during the yesterday's session. Let me share the slide for the recap.
Wonderful. That's a good idea. Thank you for that. Good idea.
So first day we had about the concept and methodology for the strategic plan. Then focusing more on the vector control surveillance, vector surveillance and vector control planning. How do we go about that? The second day session, in fact, we started about the
Pradeep, I'm so sorry. I'm not sure if you're trying to share screen, but at least for me, it's not coming up.
Okay. One second. So is it visible now?
Yes, yeah,
Yeah.
now it's good. Thank you. It's full screen as
Okay.
well.
This
Perfect.
is
Thank you, Dr. Pradeep.
basically, yes. So this is basically yesterday's session, just to have a recap so that we will start with the total continuity. Yesterday we have discussed about the entomological surveillance, vector surveillance and vector surveillance planning. What are the basic parameters? What is the basic need? Why it is there? Why the significance should be highlighted? And what are the basic parameters? Where are the hitch which can be done in the program? What are the parameters which we can do? What is basically transmission dynamics? And there were a lot of questions on this, so we could try to explain this. And I feel that people are more satisfied because the seasonality and the other the determinants, they play a very critical role for the transmission dynamics. Then there is influence of the vectors. We have given a broad picture with the aim that all the people, they will try to look for their own area and try to update the knowledge of entomological intelligence. Then we have also discussed about the vector response. Where does it raise? What feeding, seasonality, trend, and then the response and for towards the environmental change, which the vectors, they are more so. In fact, we all know that they are the most adaptable living creatures in the world, and then their response towards our intervention measures. So all these things we have discussed. We just gave one example of the vector abundance, and then we also discussed about the evidence based, how we generate the evidence to justify our proposal, whatever we are projecting for elimination, achieving the target, and trying to convince the administration, the political commitment, because that is very, very important. What is the role of entomologist in overall surveillance program when it comes after the case is detected, early stage, late stage, and the I mean the different settings also. How do we do the survey? When we have gone in detail for the vector surveillance, we have given for baseline survey, we have given for fixing the sentinel sites, we have given the fixing the spot check sites, and we have also given the flexibility when we can change these spot check sites, or we can take the additional sites. We have also discussed about grouping, that all these parameters, 23, 24 parameters, which we have been discussing, how you can group that. All the parameters, they have got its own significance. So which group and what it reflects. This grouping helps in the interpretation of the data. And then we have discussed about the WHO policy. The people, they ask, what is the core intervention? You have given a long list of the intervention. So we have all explained, we have discussed all these things. Then we have discussed about the larval measures and adult measures, emergency outbreak measures. We have also discussed about the 137 strategy for malaria elimination, and in that, where is the best fitted role of the entomologist and vector control, then for outbreak containment. And finally, we have also discussed that how we want the policy and planning to be converted into the resource planning, means the physical activity, how it should be converted into the financial implications, so that people who are having the competence to approve our plan, they are fully convinced, and then the whole thing is justified. Finally, we have all summarized that what are the situation analysis, what are the components, technically managerial aspects, why it is failing. That is basically a risk factors. If you don't do this, what will happen? So this also sometimes very, very important to point out in our document. And then how the planning resources and how it should be fitted in within the regulatory framework, within the approved norms with the government. Sometimes we do map only the epidemiological data. So we have also discussed that sometimes the data will not be sufficient to give a clear picture. So we should look beyond the mapping, also a stratification. And then we have to take a lot of other environmental and other human behavior factors. We have also discussed which activity, which broad methodology has got more impact on which diseases, vector-borne diseases, malaria, dengue, lymphatic filariasis. So all those plus and minus points we have discussed so that it helps for the planner, for the policy makers to take this issue. Now, with this continuation, with this backdrop, we would like to start for today's one, which is primarily, let me share. Is it visible now?
Yes.
Yes, it's good. Thank you.
Okay.
All good.
Thank you.
Please proceed. Thank you.
So the third day, it is all with the conceptualization and then knowing about what to do, how to convince. Now we have to understand about the what options actually in detail we do have in our hand. What are the tools, what is the technology, and where we would like to have some kind of innovations, especially for the micro planning, where if there is shift required, the flexibility which we have discussed. So if there is a requirement of shift, we can always justify and convince. Please make a point that my deliberation does not mandatorily say that you have to, after this, you can change it. No, you have to justify that because of these technical reasons, you can always convince your own seniors, superiors, administrative authorities. Look here, this is the case where we can have, you can utilize these tools. So these kinds of deliberations will definitely help to you to update your capacity, strengthen your capacity, and justify as and when the time requires for you to change or have a shift in the micro planning. Now the whole thing has been divided into we should go for tools, then option, then flexibility, and what is the situation-based application. We know that there are some vector surveillance tools and there are some vector control tools. Right from the beginning, I have been emphasizing that entomologists per se, if they are not doing entomological work, we don't consider them as an entomologist. I may be having MSc in zoology, entomology, or MSc public health entomology, but if I am doing a service in bank, I am not contributing to entomology. I am an entomologist by virtue of my qualification. But in the program, similarly, if entomologists are recruited and they are working for something else, they are not generating the data which is required for the program for entomological indices. So vector surveillance tool has to be clearly understood by an entomologist who is working in the program. The vector control tools are for those people who are actually implementing the program. They are utilizing whether it is spray, whether it is long-lasting insecticidal nets, whether they are using larvicides, whether they are helping for environmental modification or manipulation, they are utilizing biological control. So these kind of tools are more required for the implementers, and they're interchangeable, and entomologists can become a vector control expert, and vector control expert can convert as an ad hoc basis, can convert himself as an entomologist. But the person who has got training pure in entomology, he knows what is taxonomy, what is anatomy of the insect, what is physiological thing, what is resistance mechanism. So all those micro detailing, simply collecting the mosquito and dissecting, that is not the total entomology. With this, we have to understand what are the vector surveillance tool and what are the themes. Now, yesterday we have given a little bit glimpse of this grouping when we were discussing about the grouping of the parameters. Now, if you see the group of entomological indicators, when the entomologist does this survey in the program, first one is adult vector composition, where we go during baseline survey, and then there is a routine survey when there is a fixed interval. We try to collect the mosquitoes and then try to see what is the species available in that area, what are the occurrence of this species. Once we know that, okay, this is the area where we are getting it, then we will try to monitor how to monitor the changes in different seasons, in different months, at different frequency intervals, whatever we have decided. So we look for the density. Density is always a relative term. So we try to collect and then we try to explain it that, okay, one person in 1 hour, how much the mosquito he will collect or she will collect. So what is the uh, relative density for per hour, per person, and then in across the time, monthly or quarterly or annually, what is the trend analysis, whether there is an ups and down or there is a static or there is an increase or there is a decline and increase. So whether it is like undulating or there is a variation or this is a straight line. So we have to see all these things. So these are the two important parameters: the abundance, occurrence, and the density for the adult vector composition. So the procedures are different to do it. You can go for hand, you can go for spray sheet collection, whatever it is. Then adult vectors behavior. To know the vectors behavior, we collect the mosquito, try to take the blood what the mosquito has taken and try to put it with the ELISA test. Then we do the testing and then see what is this human blood index. So out of total percentage we calculate, we say that how many mosquitoes are actually they have fed on the human being. That will give the very good parameter that what is their behavior, where they are preferring human blood or they are preferring. So primarily they are anthropophilic or zoophilic. If the percentage is higher towards animal, they are preferably on zoophilic in nature. Or there may be another reason for interpretation. We will come later then. Then we also see for the behavior, what is their biting time, biting rate, time, location. In terms of location, what time they come to bite, evening, midnight, early morning, or early evening, and then what is their rate per person per night, what is their biting. In fact, we do study the biting terminology, but for biting rate, we try to collect as a landing rate. We don't allow mosquito to bite. And that is what is major change from the earlier policy to the new policy. Location, we try to see whether they are coming indoor when the people are resting inside and they are biting. That is what we call it as an indoor rester, or endophilic and endophagic, or they come indoor, take the blood, go outside. So endophilic and exophagic. So indoor feeder and outdoor rester. If they are not coming inside and out, they are only taking blood outside and they are resting also outside, they are exophilic and exophagic. But if they are coming, they are biting outside and they are coming rest to take rest inside shelters, so then they are exophagic and exophilic. These are the combination, permutation and combination of these two parameters. Then we have to identify the resting locations actually, whether they are actually outside rest or they are inside rest. So these are the parameters which when you study these parameters, you can group into the adult vector behavior. These are the indicators, very, very important indicators. Then, when you try to give your intervention measures, you are putting insecticide spray or you are putting a long-lasting insecticidal nets for the adult. These are all the adult behavior. So, whether there is a development of any resistance, what is their response to your insecticide spraying? How long they have developed the resistance, what is its frequency, what is the total resistance status, what is the intensity mechanism. Yesterday also I was trying to explain, today it will be explained much in detail, that intensity of resistance, it is 5 times, 10 times more. So these are all the parameters which will give the behavior pattern of the mosquito vector response basically towards your insecticide measures. Immature also we try to do, whether habitat is available, That is what is the area is receptive for the mosquitogenic condition, where there are good places where mosquito can breed, female mosquito can lay the eggs, and whether those areas, by looking with the eyes only, you can see, okay, with the knowing the behavior, that which particular margin, which particular shore, where vegetation, without vegetation, overhead tanks, coolers, whether they are available. But actually, whether you have got mosquito breeding there or not. Suppose there are 500 such water bodies and you have found nil. So the occupancy is zero. Occupancy by the mosquito to lay the eggs, that means it. And then if there is an occupancy, if there is a, like you have seen for the adult species occurrence. So if there is a larval breeding, mosquito is breeding, laying the eggs, and you are seeing the larvae, then you have to measure their density to know the trends, that how the breeding is coming in the area. It is picking up, which season it is not picking up, it is going down. So this also you have to take. That is what is, we have to measure larval density. There we told that it is per person per hour. That is a relative density. So here it is per dip. You take the larva, collect the larva, take the 5 dips or 10 dips, whatever it is, minimum 5, and then try to take the average with different breeding sources wise. So you take that, okay, this particular breeding source, this much density is there, is recorded in the month of January or February. So you can assess that your measures, vector control measures, larviciding measures are working or not, or there is a breeding in that. Then, having understood all these parameters, which is in routine done for the mosquito measurement, mosquito density occurrence in that area, your aim is to achieve the malaria elimination, to bring down the zero transmission, zero indigenous case in any area. Now, if you want to do that, there are two things very important. Parasite should not be there. The active transmission should not be there. For active transmission, long-lived mosquito which are having sporozoite in their mouth, in their salivary gland, they should not be available. So there should be complete absence of sporozoite bearing female inoffline mosquitoes. Now to see that, we have to do the dissect the mosquito salivary gland. We have to see what is the sporozoite rate. So percentage of sporozoite bearing mosquito positive against the total mosquito observed, that will give this information. So sporozoite rate is basically a proxy for transmission. This is very, very important parameter. And if the malaria transmission is going on, and if some entomologist comes and says, sir, we could not get mosquito, there are no case, but I do not know why the malaria cases are occurring, why the infant parasite rate is there, it means our entomological survey is not adequate. It is not done by the capable capacity people. They have not been able to search the proper place. There are many, many examples like this. I can go on quoting, but this is for the formulation of the surveillance draft plan, strategic planning. So we should understand that sporozoite rate is very, very important parameter to declare an area whether there is a transmission, is stopped completely or not. So that is why it is known as a proxy for transmission index. There are two more index which is very important, entomological inoculation rate. But as I told you, that entomological inoculation rate, it takes another 2 hours to explain, because entomological vectorial capacity and entomological inoculation rate, it can be done in a research mode, it can be done by the scientists, it can be done as a study in some area, but across the country, across the states, across the district, by the program people, it will be little difficult, because you have to collect many, many parameters. Then you have to go and then you have to do with the log probit analysis. You have to take the log value of that, and then you have to convert into entomological inoculation. But by and large, entomological inoculation rate, one mosquito, infected mosquito, he produces how many victims, how many these things. So how many times it inoculates per day or per year or per month, you can calculate relatively. That is what is entomological inoculation rate by a single infected mosquito. Receptivity is another important parameter. Receptivity is the area specific, that if you have seen the area, whether your area, that is the first concept. Physical watching will tell you that whether the area is congenial for mosquito breeding and mosquito proliferation or not. Then you have to go and check all these other parameters by routine survey in this area. And if you have already achieved the zero indigenous transmission in an area, then you may not get adequate mosquito. If real picture, you may not get parasite, malaria parasite, everybody will be healthy. You don't have the mosquito to transmit, female mosquito, you don't have the gametocide carrier, you don't have the infected mosquito, but the area is receptive. So you have to be very, very watchful for, especially for the larval breeding. If there is a larval breeding of anophelines and the area is receiving some vulnerability issue comes in that population migration, the receptive area will become a focus for allowing the case to establish and then flaring it up in later on. So this is very, very important. And as yesterday also I was telling, if we are having multiple infection or multiple influence of the malaria vectors, if an area there are, say for example in India, or we know that there are total out of the vector species, malaria vector transmitting in your region, in your area, if the area is under the influence of more than one vector, so all these parameters should be done and explained for all the vectors separately. Don't club and say that malaria vector density is like this. Never. Malaria vector incrimination rate is like this. No. You have to tell that sporozoite rate for Anopheles culissifacis is this. Sporozoite rate for Anopheles stephensi is this. So those vectors, sporozoite rate for fluetalis, for minimas, for biomai, it is all different. So all these, that is what is the important caution. Now, how do we go about that? If you go look for the techniques, we have discussed about the landing collections, biting rate. So if we talk of human biting rate, after the adult vector composition, if you see the human blood index and human biting rate. So if you discuss about the human biting rate, whatever mosquito you have collected, either with the hand or with the if they are full fed, you can just take the blood and go for the human blood index. That will come later. But if you want to know the biting time behavior of the mosquito, nowadays, as I told you, that human biting collection is not allowed because of ethical reason. We do not know that whether they are inoculating malaria parasite or some mosquitoes are carrying some virus and they are inoculating that also, like dengue, chikungunya, and JE. So we don't take a risk in the program. So we don't allow the landing, and that is what WHO has recommended, that biting collection is not allowed. It should be termed as a landing collection. You can, but this is so expertise work that when you are collecting, you should not allow the mosquito to bite. Number one. So when the landing collection is done, hourly basis, we know that mosquito starts biting late evening and then come to the, so whole night, because primarily they are all nocturnal biting, not for Aedes we are talking for malaria vectors. So primarily they are all nocturnal in nature. So we start at 6 o'clock. Nowadays, in fact, with the late sleeping habits and more work culture going even till the rural areas, so most of the people, they don't sleep at 6 o'clock or 7 o'clock. So you may not get mosquitoes between 6 and 7, but you, for 12 hours period, you start your work at 6 o'clock. So 6 to 7, 7 to 8, 8 to 9, 9 to 10. Like that, you divide the 12 hours hourly basis. And one person starts collecting. The moment the mosquito you feel there is a torch on the head, or you have to torch the other person holding. So flash of torch, minute to minute, second to second, you are just putting it. And then you are seeing it. The moment you see the mosquito landing on your leg, you expose your parts, leg parts basically, and you can just suck it. So that is what is human landing collection. So If there are 2 or 3 people at different places, so you just pull the mosquito and see that in 6 to 7, how many mosquitoes, out of that, how many anophelines, out of that anophelines, how many are malaria vectors. So you can work out that this particular time, and then you see that 6 to 7, 7 to 8, what time maximum malaria vectors are landing. So they are trying to come and bite, pick, suck the blood, but you have taken them. So you can understand that what is the biting time. And then within the biting time, suppose it starts picking up. It starts picking up from 8 o'clock night, and then you see the peak coming at 11 o'clock or 12 o'clock, and then suddenly the mosquito density goes down. So their habit is going down. So you can always say that biting time starts from 8 o'clock to 3 o'clock morning, but the peak is around 11, 11:30 or 12. So in that particular time, whatever mosquitoes you have collected, that will be much more in the number. There is another method, human baited trap, which is again not allowed nowadays because you can't put human as a bait. So we use animal baited trap. So animal baited trap, we collect, it will not be as a human biting rate, but it will be a biting rate of the anophelines. When is the peak biting time? When is the start of biting time? So that again in the trap we try to collect on hourly basis, or we can collect whole night. We can go and do the collections, and then that will come per trap per night for the animal biting collection, whatever mosquitoes you have got. And then from there, maybe that they are somehow entered partial, and then they have taken partial blood of human, partial blood of animal. So when you analyze that human blood analysis, it will give for both bovine and human. You also do human odor baited trap with this. One famous is carbon dioxide CO2 weighted trap. CO2 is known attractant for the mosquito because when the CO2 comes out of the nose, so CO2 has been taken. So there is a cylinder which slowly, very slowly it releases, and the trap comes like. There are some light traps also, which are also taken, CDC light trap. The only problem with the CDC light trap, you can collect the mosquito, you can identify them. That sometimes, if the fine speed is not regulated, the mosquito may get destroyed. Mosquito, definitely all the mosquitoes will be killed. No mosquito will be alive. But sometimes the mosquito, like wings are gone or legs are gone. So it becomes sometimes difficult to identify also. But it will give definitely an idea that what is the time when the mosquitoes are coming up. We have to be very, very watchful for fixing the light traps, which area we are doing and which area we are trying to collect the mosquitoes. Another important parameter, human blood index. You can squeeze the mosquito abdomen when you collect, put on the filter paper, and there is a system of filter paper. You fold the filter paper different angles and then make it, and then one is small boxes. You just go on taking the filter paper. It is basically a round filter paper, and then you just go on folding. Then 2, 4, 6, 4, 12, something like 4, 8, 16, then 32, 64 like that. And then every column you just go on squeezing mosquito from the abdomen side, and then whatever blood comes, you just take a round spot. Then those things are cut and taken the analysis, molecular or enzymatic analysis of the blood-fed mosquitoes, and that will give, with the serological test, it will give that which blood it has taken. Then you can make out the anthropophilic index. Resting and feeding behavior, whatever total collection you have done in the area, from the indoor and outdoor, you have to segregate the collection. You have to record separately that this is from the human dwelling, this is from the cattle shed, this is from the mixed dwelling. And then you can understand this is from the indoor, this is from the outdoor, this is from the bushes, this is from the resting area. Sometimes you can get between there are many houses in the rural areas as a mixed dwelling. See, part of the house is for the cattle and part of the house is for the human dwellings, and in between there is a gap. So you have to use your own wisdom, whether you are taking it as a mixed dwelling or you are taking it as a human dwelling separately if the gap is more. So you can just record it and then identify that, okay, the resting collection and feeding behavior put together, it will give you the exophagy or endophagy index also, your endophilism and exophilism, and the feeding behavior, everything we have to, they are all so interconnected. I just want to say. And indoor collection, definitely, as the name says, we are doing only indoor collection. We don't mix the tubes. All the tubes which are transferred, they have got pre-decided labels are already given. And outdoor collections, for exophagic or exophilic, we always do the water. Outdoor collection only, they are taken into account. Another important parameter which we were talking about the adult vector's response is the resistance, the behavioral patterns or avoiding pattern for the mosquitoes to avoid the tolerate the insecticide, whatever we are using. So we try to measure the physiological resistance. Not forget about, but we are not going to discuss the whole resistance mechanism here in this chapter. There is an avoidance, vigor tolerance, and then there is a mechanical. But here we will be talking about the physiological resistance, where we actually expose the mosquito against the insecticide concentration, known concentration, and then give them exposure for a limited period, whatever is decided. And we usually follow the WHO standard procedure for 1 hour for malaria mosquitoes. So 1 hour we try to put a paper with the impregnated with the insecticide concentration, known concentration which is known as discriminatory concentration. And the discriminatory concentration is actually double of the concentration what is able to kill the 100% LC90 or LC100. So the moment you see in the lab that this particular concentration, 100% mosquitoes are being killed, you just double it and then make a discriminatory concentration. And that paper is supplied, is standard paper, impregnated paper are supplied through the WHO from the Malaysia. And then one second. Talib Hussain Joya. Please. Thank you. So the video was on. So what is happening that we try to measure the mosquito exposure and see the mortality after 1 hour, whatever paper has been supplied, and they are known as exposure tube. If the methodology is very standard, so we have a full-fledged kit of measuring it, and there are tubes, the exposure tubes, and then the control tubes with the yellow marking, and then green color, the holding tubes. So you put in the red mark dots, you put the insecticide papers, and simultaneously with the control papers. So you expose the mosquito for 1 hour, again you release the mosquito to the holding chambers, allow them 24 hours more after the 1 hour exposure, and then see the mortality, that how many mosquitoes have been killed. And every tube you are exposing 20 or 25 mosquitoes, female mosquitoes. So you just see that how many mortality. So this mortality you try to, this is basically a physiological resistance. Physiological resistance we are, and when we are fixing the sentinel sites, we try to take this consideration that whether these things have been done in that particular area, or these things are available, whether the epidemiological stratification, if epidemiological stratification has not been done, we will not be able to locate which is high burden area, or which is low burden area, or which is meso burden area. So we will be very difficult for us to go for case-based stratification. Once we know that this is very high reporting malaria cases area, we will definitely go for that area. And then we should have the data for the vector distribution. We should have what type of intervention, whether the area is under LLIN, whether the area is under insecticidal spraying, whether the area is without any intervention, and then what is the insecticide pressure or resistance status. So all these things, old data, old parameters we have to take, and this is very important when we are trying to select the sentinel sites. Usually, if anything is ignored, our fixation of sentinel sites, I mean, the statistical people will say that you have to do only on random number basis. But that may not give a correct picture for different. So for that only, these are all the factors which will lead you for cluster-based randomized selection. So all these parameters you take, make a cluster, and then in that cluster, yes, you can take any of the sites. Now, when you are measuring the resistance, you may find that mosquitoes are falling in, the resistance status is falling in certain criteria. What is the percentage mortality you are measuring? But that is also grouping has been done, that less than 90%, between 90 to 98% and 98%, 97% above. So I'll come to that. And this resistance mechanism, which most of the time program people, it will be very difficult for them. We have to take the help for the metabolic resistance or cuticle. There's a mechanism of resistance. So we have to check for the take the support from the some research organization or some academic institutions. For larval, we have to, as I have told you, that water bodies presence has to be mapped, and then frequency and timing of survey depending on the length of the.
And then that's also people are not relating to this.
Mr. Wilfredo Ore, can I ask you? Oopsie. Sorry, there was, yeah, thank you for muting yourself.
One second. I was just about to tell that we have to take the support from the research organization or the academic institutions to support us for.
Mr. Abdul Hamid, can you mute yourself, please? Abdul. Thank you.
Thank you. Thank you. So I was just emphasizing that for certain advanced parameter for metabolic resistance or the mechanism of resistance, we have to take support from the research organizations or the academic institutions who are competent, because program people are more busy on the implementation side, and some of the entomological parameters which are taken, they are taken just for the important purposes and important parameters based on the priority things which are required. Sporozoite rates and entomological inoculation rate, I have explained that they are why they are proxy for the transmission. That when you are actually measuring, the sporozoite index, it gives the rate of infective mosquitoes present in your area, which is very, very important. Because if infective mosquitoes are present in your area, you just can't claim that you are able to interrupt the transmission. Because if vector is carrying sporozoite and biting, taking blood is its need. So the moment it will take blood, it will inoculate. That is, by default, this sporozoite will go inside the human body. So you just can't claim that you have been able to completely, just only because of numbers you are not getting. Maybe your surveillance system is not adequate to capture. Maybe your case notification from many agencies. There are government agencies, there are private agencies, there are hospitals, there are dispensary. Who is reporting, who is not reporting? 100% absolute insurance cannot be given. So if you are getting even a single infective mosquitoes, it will be very, very difficult for your claim to establish and generate the evidence, and your claim cannot be verified. That is why it is known as proxy for transmission. Then entomological inoculation rate. I have already explained you that this is measured basically for intensity of malaria parasite transmission. What is what is required is, it's basically a type of mathematical modeling and mathematical calculation. That's what I was telling, that it is basically number of infective bites received per person in per unit of time. So if vector mosquito is infective carrying the sporozoite, how many persons it will bite in a unit time? Because every bite in malaria, the mechanism is entirely different than lymphatic filariasis or any other disease. Every bite, the mosquito has to put the saliva inside to as an anticoagulant, and then the sporozoite, the salivary gland containing sporozoite, they will be inoculated. So it will, by default, it will go. So that is why inoculation rate, entomological inoculation rate is another important parameter, but it needs lot of strengthening of the capacity and the all the kinds of facilities to be provided, so that they can go in the night time, they can right from transportation, capacity, HR, so many things are required for measuring the entomological inoculation rate. So better you take the help of support of the technical organizations who are supporting as a research or as a scientific or as an academic institutions. Receptivity, I have already told, and WHO is still evaluating. I told you there was one paper which has been published, very nice paper on the receptivity. It has given history of receptivity, how we started considering which is the area is receptive, and it has given right from 90s. The paper has published, and I'll try to share that paper with Leo or Ulan so that they can put on that. So very nice paper. It has come in a very peer-reviewed journal. Frequency. Frequency of surveys is very important, and many program people, they ask this question. Many of the program people, they ask this question that how, what, and how many times we should do the survey. Whether one time survey, because they don't have. When there is a resource, you don't ask such questions. But whenever there is a resource constraint, you ask these questions. So when you are asking the question, I am trying to simplify that it depends on the available resources. That is the first thing. The second important point is that when you are having some resources, you have to assess what is your transmission season. That's what the first day, second day, I am showing one graph every day, that spray graph, that what is this? The transmission season starts picking up from May and last till September or August. So 5.5 months or 5 months the transmission season. So you have to see that, and you have to do one survey, pre-transmission, one survey, post-transmission, one survey during the transmission. This is very important point. Otherwise, you may not get the correct picture of your samples. Frequency and timing should be standardized so that you fix up, say, for example, a 5.5 months transmission. So one you have fixed up in early May or April, you have fixed up one somewhere in June or July, and another you have fixed up somewhere in October, post transmission period, or November. Fix it. Fix the timing so that there is no bias. So sometimes you go in February, sometimes you go in March, sometimes you go, sir, we did not get the mosquito, so we have gone in February or March. You can go, but when you are doing trend analysis, you should not club that data with this data, because trend analysis will be faulty if you club randomized trial with the sentinel trials. So that is what is given. And if you don't have even that much resource, you can always take that whenever there is a peak, that this will come, peak vector density. You have seen the routine survey, baseline survey, you have done preliminary survey. You know that which season the density is increasing or density is coming to the peak. So that particular 1 month must be there so that you get more number of mosquito and see the trend, that every year or in a period of 5 years, what is the trend analysis? Whether if the peak by mosquito density was coming in the month of July, and you are doing all these 5 years program, strategic planning. So now your July density has gone down. Your peak density does not touch to 50 per man hour or 30 per man hour. It always remains at 10 or 8 or 9. So this is very, very important parameter. Usually we do on weekly or monthly basis. For larval density monitoring, we do on weekly basis, and for adult vector density, we do on monthly basis. This is very, very important. So, but it all depends on the resources. Now, another important chart which you want to see, if you just see it. We have discussed about the indicators, occurrence. What is outcome of this occurrence? If you want to see this technique, if you want to see what for you have been doing, occurrence will say that whether the adult female vectors are there or not after when you started, and what is the situation now. So that is why the trend analysis is done. And this is done because you have to see, you have to be taxonomist also. You cannot ignore taxonomy for identification of the Anopheles vector species, because there are slight differences for all these things. Then another parameter is density. Density is what I have told you, that if there are 5 people, they are collecting, they are spending actual time, everybody is spending 30 minutes. So 30 minutes into 5, total actual time spent for collection is actually 150 minutes. So in 150 minutes, how many total mosquitoes, vector mosquitoes they have collected? So 60 minutes. So pull together and then take out one individual vector species and divide by. So calculate for 1 hour how many vectors are there. But mind it, don't use multiplication factor. Use only the factor, dividing factor. So if you have to collect per man hour or per human hour density, your total time spent in collection must be more than 1 hour. So if everybody is spending, suppose there are a team of 5 persons and everybody is dividing themselves, and every 5 persons are going, and you are spending 15 minutes in one structure, and you have taken 10 minutes, 10 structures. So if 15 minutes you are taking, so individually you are actually spending for collection 150 minutes. So 150 minutes is actually 2.5 hours. So 150 minutes, one person is spending in 10 structures for collection, and there are 5 people. So 2.5 hours, 5 people are actually collecting 5 into 2.5. It becomes roughly 12 and 12.5. Then you can divide that in 1 hour, how many total mosquitoes. But don't say that you spend only 30 minutes. If one person has gone, if only one person has gone, you just collected 10 minutes or 15 minutes and multiply by for 60 minutes. Please don't do that. Second one is human blood index. This is actually we calculate as in percentage of human blood fed vector. So out of total mosquitoes, total vectors collected, how many, what is the percentage of human? That is what we call it as an anthropophilic index. So we just take we do test for human blood, we do test for bovine blood, we do, and whatever serological samples or sera we have got. So we try to put so many things, but we take out human blood and what is the human blood. This will actually tell that what is the preference feeding in this particular area for the vector mosquitoes for human beings. So the chances of getting infected by the mosquitoes will be more. If the area is with the influence of zoophilic vectors, the chances probability will be less. Like for example, one I give you, Anopheles minimus. It's a highly anthropophilic. So even a small number of mosquitoes, they will be able to transmit malaria in that area very rampantly than Anopheles culissifacis, which is primarily zoophilic in there. So this is what is the importance of this. And so the outcome is percentage of human blood fed vectors. Now biting time, we just calculate number of female vectors landing or biting in different hours. So we know the peak biting time, human biting rate or peak biting time, we try to calculate in the night. If you see the old literature, you will see. But nowadays, people are not going. People are doing some kind of error also in this particular parameter, that hourly collection, they only go for 15 minutes and come back. So if you have gone for, say, for example, 8 to 9 period, 8 p.m. to 9 p.m., you have just gone for 15 minutes and the mosquito is coming at 8:50 p.m. and you have fixed up your time for 15 minutes, 8:30 to 8:45 or 8:20 to 8:40. You will not get the mosquito. So your whole thing is faulty. So please don't do such kind of error. If you want to see detailed guidelines, it was very nicely given in 1972-73, the WHO Practical Manual of Entomology. This is still available on the Google. They have not removed it. And there are two parts. Very nice explanation. One step by one step they have given. And there are many other books also, many guidelines we have. I mean, our India guidelines also we have explained very nicely all these things. Then resting location indoor and outdoor. What it gives the proportion of female vector mosquitoes, malaria vector mosquitoes. What is indoor versus outdoor? This again gives, if your people sleeping habit is indoor and there is a more proportion is high for indoor rester or indoor biter, then the probability is high for contracting malaria. If the people are having outdoor sleeping habits and mosquitoes are indoor, probability become less. But if the outdoor feeding mosquito, outdoor resting mosquitoes are there, vector mosquitoes, and people habit is also outdoor, then the synchronization, and what happens? This kind of endophilic index or exophilic index, this increases the probability along with the sleeping habits or resting habits of the people during their biting time. So peak biting time, resting habits, biting habits, landing habits, this if you mix together, what happens? That it gives a synchronized interpretation that look here, because these are the things which are helping, which are supporting, these are the parameters and this is the factor. Because of this, this area is highly vulnerable for this. There is a probability of malaria transmission. So even one single mosquito, infected mosquito may create a havoc. So please be alert on this. This is what is the significance of entomological surveillance. Come to resistance monitoring. Resistance monitoring is basically, we have got 4 indicators. One is frequency, one is status, one is intensity, and one is pattern. When you want to understand about the frequency for resistance monitoring, basically it tells you what is the percentage of adult female alive after exposure. You are giving 1 hour exposure. Only 1 hour exposure in an exposure tube, red dot marked tubes, which are having insecticide impregnated paper. So out of that, how many are killed? That is what the frequency of resistance. And this is their exposure time, 1 hour. Please remember, it is not 2 hours, it is not 3 hours. It is different for some other insecticide and some other vectors. But for malaria vector, by and large, for program persons, it is only 1 hour. The status, resistance status, we do expose them for 1 hour and then transfer those mosquitoes to a holding chamber, which is only with the plain papers, not with insecticide impregnated papers. So they are given rest period for 24 hours. It is actually 24 hours is not included in that 1 hour. So actually total time for resistance monitoring is 25 hours. 1 hour exposure added plus 24 hours. After 24 hours, see, suppose today 10 o'clock you have put the exposure, 11 o'clock you have taken out, 11:10 or 11:20 you have transferred them and then set right the, and then fix your clock at 11:20 next day morning. So when you are counting there, you are seeing how many total number of mosquitoes they are killed, take out the percentage. Now what is important is WHO has changed their criteria, and the recent criteria is that if 98% or 98% more than mortality, means mosquitoes are killed more than 98%, then the insecticide is effective and mosquitoes are susceptible. If the mortality is between 90% to 97%, So there is a probable resistance, there is a possible resistance. We have to again repeat and confirm it. But if your mortality is less than 90%, means mosquitoes are alive. So it means they are resistant. Insecticide is not effective. This is very, very important parameter which we have to understand very clearly. Then we go for the intensity. This will take some time. Dr. Leo, is it the time to break? Because this intensity of resistance will take some time.
All right. If you suspect it's going to need a little bit of time to explain, let's break for 10 minutes then. Reconvene in 10 minutes, please, everyone. To you.
Right.
Thank you so much.
Right. Thank you, Leo. Thank you very much. And again, taking the clue from the previous things, we were there on the frequency and status, and then intensity is another important things. You know what has happened? Intensity test was not there earlier in WHO guidelines for resistance monitoring, and many of the people still they are not using intensity bioassay test. This is basically bioassay using the bottle, which has impregnated the inner layer of this, which is impregnated with the insecticide. This resistance is actually taken into account. This test is taken into account for the purpose when you have less reliable epidemiological data. Earlier, the concept was that you observe the resistance and then you see the epidemiological impact also. Even with the resistance reported in your tube test, which was earlier I have told you, if you have taken that and you are getting epidemiological impact, you can continue, because actually you are forcing the mosquitoes, you are capturing the mosquito in a very small tube, and then you are taking them and then trying to study the resistance, the mortality. So you are actually giving a forced exposure to the mosquito, which may not be possible in the field condition, in the wild condition. As we progress further, everywhere epidemiological data, epidemiological surveillance system, it is not as robust as the primary health care should be. There may be gap in reporting, and that is why there are many publications which are coming that your reported cases are at this level and your estimated cases with the mathematical modeling and estimated is at this level. So there is a huge gap between estimated number of malaria cases. How do you justify that? All these concepts were taken. How do we interpret and decide based on only resistance monitoring data, whether we have to go for alternate insecticide or not? So intensity test was brought in with this concept. And, basically you are testing at, say, for example, 4% concentration. You make it 20% concentration, you make it 40% concentration. So at higher dose, you are seeing, even if higher dose mosquitoes are surviving, it means the resistance is beyond doubt, it is confirmed. And that is what is intensity resistance. If you look on this table, I have tried to explain little more simple terms that if you look for this, we go for this. Once you get for the first one, the resistance tube test, and if it is resistance 90%, then what we do? We expose them 5 times more concentration. We expose them at 10 times more concentration. So the intensity concentration, the criteria remains the same. But if 5 times or 10 times, if 10 times exposure with more than 98%, it's a high resistance. Suppose the mosquitoes, they do not die, then definitely there is a resistance. 101% sure. So if 98% mortality after 10 times exposure, less than 90% after 5 times exposure, then there is a little moderate. So the first criteria is that if 98% mortality, less than 98% mortality, it means Mortality is, I mean, the mosquitoes becomes susceptible if more than 98% mortality. So if mortality is less than 98%, then the exposure 10 times, also the resistance is status is very high, intensity is very high. But if it is more than 98% mortality, but less than 5 times exposure, I mean 5 times more insecticide concentration, it is less mortal. Some are surviving. So then there is a moderate kind of resistance. But if in both the cases, 5 times exposure, 10 times exposure, it is more than 98% and less than 98% after 5 times exposure, then the resistance is at the low level. This is very, very important point which we should understand. If 100% resistance is established, even with the 5%, 5 times expose more insecticide and 10 times more concentration, then 100% resistance is declared and you don't have any other option but to go for a alternate insecticide. This insecticide is completely becomes ineffective. This is very, very important point which you all must understand. Now, the mechanism part, as I have been telling, that we have to take support for mechanism, molecular or biochemical testing. It is done. It is done. We in the program people, I have not seen anybody who is doing in the program people because they are very busy. You have to see cuticular resistance, or you have to see the biochemistry, physical molecular resistance, which particular factor is taking. So, or KDR gene, where it is responsible, what is the knockdown resistance. So these are all different parameters which we cannot take in the program. Up to this place only we have to make. We cannot go more technical details in our strategic policy framing. So the whole presentation, whole deliberation is, I am restricting to only strategic planning and strategic formulation. You cannot give that, okay, we will do all these things and finally you will end up, you make a financial resource planning and then you say, we did not get so much things and we just thought that we will do and we will not able to do. Please do not land up this problem. That is why I am restricting to those technical details. I am not trying to explain which is not required for strategic framework or strategic planning. Because the methodology of resistance monitoring, intensity monitoring are entirely different. Only thing we can always put in the strategic planning that if we get the resistance, we will go for intensity test resistance monitoring, or we can go to see the mechanism. We will request to the academic institutions or the research organizations. For that, some support will be required, which we will be doing. And always there is a buffer amount kept in any strategic plan. So from that we will meet. Now, coming to the larval habitat parameter, when we see habitat availability, that is basically receptivity for the breeding potential, whether the area is having a lot of potential for the mosquito breeding, especially for the anophelines, fresh water, slow moving streams, shaded streams. For urban side, it is the overhead tanks, stored waters, coolers, desert coolers, so many things. Building sites, industrial township, water aggregations, water storage in the containers because of deficient water supply, regular water supply. So these are the things which promotes water stagnation and water storage. So the area becomes receptive. So habitat availability is one thing. And then habitat occupancy. What is the outcome of the habitat occupancy? That we will see whether there is a breeding or not. So the habitat is positive or habitat is negative. So the breeding source is positive, breeding area, breeding potential is not. If the breeding is less, the breeding potential is less. If the breeding is high, out of 100 breeding sites, if you get 90 positive or 80 positive, it is definitely high. So breeding potential. So larvae present or absent, larvae and pupae both immatures. We cannot see eggs inside very frequently with the naked eyes. So the area is positive for breeding or not. This will tell that what and how we calculate that number of total number of 100. I gave you the example. Suppose the 80 are positive, so 80%, 20 are positive, 20%. So having done this, if there is an occupancy, then we will go for the third parameter, larval density. Larval density is number of immatures or the vectors which are collected from individual habitats. If you are collecting from the well, you are collecting from the shore margin of the river or pond, you are collecting from container. So different type of breeding sites you list out, and then you see, well, what is the number of larvae you have collected. If there are 2 people they have collected and they have used 5 dips in everything, so they have actually done 10 dips. So out of 10 dips, suppose you have collected 1,000 larvae. And out of 1,000 larvae, you know that how many are enough lines. So what is the density per dip? So that is what. You can segregate it. First and second separately, first, third and fourth separately, pupa separately. Then you can see first to fourth, that is an immature total, first to fourth and plus pupa, that becomes the total immature density. So you can always segregate and interpret. And everything has got its own significance. If you are getting early instars, it means the breeding is there. It means the mosquitoes are there, they are laying the eggs. If you are getting third instar or pupae, fourth instar or pupae, it means your vector control is not good enough to kill the third instar and fourth instar larvae, because third instar and fourth instar larvae or pupae, it will take some time. So 7-8 days. And your weekly larval application is also for, if you are doing weekly, it should be effective 8 to 10 days, 1 week. So if 1 week you are seeing, last week you have done and this week you have done, it means if you are seeing third, fourth instar larvae, it means your this weekly larvicidal application is not effective. This is very, very important. The last important slide for the parameters calculation is again the proxy calculation, but you must understand sporozoite rate. That is basically percentage of adult carrying sporozoite out of total mosquitoes dissected. This is very important indicator of the proportion of infective vectors present. I have just answered that some people they ask in the chat question box that whether in the low endemic area sporozoite incrimination. How do you establish that transmission, indigenous transmission has stopped? Indigenous transmission is stopping means you have not got any single case, number one, in that area during last 3 years. If at all you have got a case, it should be established that this is reported because of case history. But if the person has not revealed the entire fact, or your total pro forma is, for some reasons, it is not filled properly or the information is not documented properly, how do you say that your indigenous transmission? So their entomological support will be required. If you are doing this sporozoite incrimination rate, maybe by manual dissection or maybe by pulling the mosquito or testing for sporozoite in a pool, so that the pool of the mosquitoes is not positive for sporozoite. What is more common in LF as a xenomonitoring, or for Zika, it is xenomonitoring. So, or PCR technique, if you can do in the pooled mosquito, crush it and then see for the sporozoite. So if you are not getting any infection in the mosquito or infectivity in the mosquito, it means you are 100% sure that the indigenous transmission you have interrupted completely. Entomological inoculation rate, I have already explained that there is no standard protocol for this. Many of the scientists, they have done during and they have published their papers. Basically, now you see the complex thing that what is given in that this number of infectious bite by the adult female per unit time or unit time is mostly taken per year. Now, how it is calculated? That human biting rate, that is per night. Sporozoite rate, again here it comes inoculation rate. Then human landing catches or vector density. by default we take vector density and human density and sporozoite. So all these three multiplication factor. So the most important point is that of all the three becomes the entomological inoculation rate. And this is reported basically per year. You cannot go for per night, but you have to calculate per night so that you can do. But yearly calculation is the best one. Whatever the documents has been published for EIR. But then it is almost similar to the vectorial capacity, but there is no standard protocol. This is basically a research people, they may support you if you want to generate some documentary evidence on that. Receptivity, as I told you, I'll be sharing that paper, that very nice paper, which has come 23 only. It was published 22 or 23. That it will give that the area is actually very creating a congenial atmosphere for the mosquito. The vector is competent, there is a suitable climate, and the human population is susceptible. If the vulnerability is added, if the population migration or establishment or imported case, everything, how many places you have to track. But when you are landing in a interrupted situation, post-elimination phase, you have to be watchful. And this is more simpler that with the just visual appearance, you can say that this area is receptive. Then there is a mosquito prevalence, that the first one is the abundance or the density will tell you, regular monitoring by the entomologist, it will tell you. And then climate, you can always take a record of that what is the relative humidity, what is the temperature range, and what is the rainfall total in this area, what is the season. That particular season, you can always have the vigilance, very rigid and very strong vigilance you can do, so that you will be able to tell that this area is. But I have come to know that the WHO is drafting and they will come out as a receptivity index, when that will be a statistical parameter for to monitor. What are the factors we have to take into that account? What will be the weightage for all those things? That will come later. Now, what are the surveillance tools? Basically, what we use. We know that for mosquito collection, adult mosquito collection, we do the resting collection at different surfaces, because the mosquitoes, anopheline mosquitoes, they sit with the walls. So what we do, we use indoor and outdoor collection with the hand aspirator. Hand aspirator is not like just like a suction tube. There is a tube. There is a rubber tubing also. There is a glass tube in between. There is a barrier which is having a filter, so that mosquitoes, they are not coming inside our nest. This, the resting collection, hand collection, we get density, resting habit, distribution, physiological age of the mosquito also we can dissect. We can dissect the ovary and see if how many times they have laid the eggs. First thing is that whether they have laid the eggs or not. So we see the tracheal skeins. If it is skein is uncoiled, it means they are pairous, they are not nulliparous. But if after egg laying, every time that ovary that shrinks and there is a knot formation like this on the ovary. So if you see 1 knot, 2 knot, 3 knot, so you can calculate that what will be the gonotrophic cycle and at how many intervals they are dealing with the species anophelines. So go on adding 1.5 day or 2 days on an average. So you will see that what will be the physiological age of the mosquito. Survival rate, we do collect daily survival rate, collect individual mosquito, put in a test tube, and then leave it as it is. See what is the percentage of mosquitoes being killed on every day naturally. That becomes the daily survival rate average. Blood mineralosis, I have already discussed that human blood index. We usually collect during morning time and evening time, dawn and dusk, between 6 a.m. and 9 a.m. and then 6 p.m. to 9 p.m. Because of this, we do take this. Because in the daytime, the moment the sun rises, the temperature increases, mosquitoes, they will try to hide themselves in the creeks and crevices, so it will be difficult to collect them. And then being a nocturnal, they will take the blood and they necessarily they had to take some rest in the They need to take rest on the walls to digest and get their body lighter. So they will take the rest. So that is why dawn period is best for the collection of the mosquitoes. And evening time also, swarming time, when they take the blood, they may take rest immediately. So we can collect, but best is the dawn time. What we do it, we should have a suction tube, aspirator, as I told you. We should have a torchlight. We should have some test tube, cottons labeled with the cotton plugging. These are very important things. What we do for the adult collection, so resting collection, hand collection, and then for resting we do pyrethrum space spray collection. If there is less number of mosquito, we try to spray with the 2% pyrethrum extract spray in the hut, close all the openings, put the white bed sheet down below, just spray, space spray, and come back closing. After 30 minutes, we open the door. All the mosquitoes which are hiding themselves, they will fall down on the white sheet. We try to take out white sheet slowly, take out and pick up one by one, put it in petri dish, uh, placed with a just wet filter paper, so that even if some mosquitoes are alive or some are knocked down or some are moribund, they should not fly off. So that their wings are wet. So we just try to count out. So this is what is resting collection. For biting collection, as I told you, human and animal baited. Human baited is biting collection is not allowed, so it should be landing collection. Trap collection, we do fix some trap, exit trap or entry trap, so that the mosquitoes which are endophilic and exophagic, so they will try to come in, go out, or they are biting, feeding inside and then going for outside resting. So the entry trap we fix up in the window. Oh, the conical, one thing is on the square side, it goes like this, and then the tapering side is opposite. You reverse it, it becomes the entry. You put it like this outside, it becomes the exit trap. So it is a window trap, then portable trap. There are portable trap which you can just move it and fix it somewhere. You can put some animal inside, or you can put some attractant killing devices. So you can use different kinds of traps, mechanical devices, light traps are there. More common, everybody knows about the light trap, CDC light trap. There are some modifications. There are bigger traps. There are attractant traps, CO2 attractant traps. Then for the larval collections, we use ladles or dippers or well net also we do. There are many kinds of photographs for this. Then in this kind of collections, what are the challenges? Challenges you see. Who will go and collect, do the entomological survey in this cooler to check the breeding? Is there any way to collect? These are all multi-storied fitted with the coolers. See, this fellow is trying to give a larval ladle dip in this water tank. There is no stairs. He has just borrowed some stair from somewhere in his eye. Who is at the rescue of this person? So these are the entomological surveillance challenges. Very, very difficult. See, and the number, so much variation. This building construction, industrial township. How many laborers, how many contractors, what kind of breeding sites? Innumerable breeding sites are created, especially during rainy season. And when they are curing, they have to collect the water and they have to put this. Then these kind of thrown away things, you see, very good breeding sites. These are all open tanks. There are many thousands and thousands type. You all may be aware about that because the audience is fully aware about this. How to do the collections, you see. These are all the collecting the animal bait. This is the suction tube. These are the torch. They are picking from the after the pyrethrum space spray. These are white sheet. The mosquitoes fallen. They are being collected, kept in the with the forceps and kept in this. These are all the window traps. You see, this is the outside. This is exit trap. This is inside house. So they are entry trap. The mosquito will enter, but it will not be able to go out. These are all the light traps, fixed light traps at different heights you can fix. This is the, see, animal trap. animal bait which is put inside. This is animal biting and this is animal bait. The mosquitoes have taken blood of this animal, and then there is a trap inside. So these children, they are insect collectors. They are collecting the inside this trap. They are trying to collect the blood-fed mosquitoes who are taken blood, and they are arresting it. These are all. This is another type of trap which is actually you try to collect during the evening time, swarming time. This is just like this kind of window trap, little bigger in size. So you just put the vehicle and go there, and so all the mosquitoes with this, they will come inside, but they will be trapped here. They cannot go out. This is only to know the abundance in this area. This is another vector control tools. You see, this is one very common, known as a insecticide treated nets or long lasting insecticidal nets. This is impregnated with the insecticide at the time of manufacturing yarn. These are all the indoor residual spray. All the 5 walls to be sprayed for malaria. These are all the screening of the houses. You see, the screening of the houses is the best tool. This is community perception and the individual response. They are taking this screening. I was seeing in one of the webinar that they have done some study in Africa. They have done some project, and they have produced a documentary evidence there how it has helped. It is also very good tools. So beside these vector control routine tools, insecticidal measures, there are some emergency measures. This is a fogging machine, handheld fogging applicators. You put the fog. This is a pulse jet technology. So there are 2 tanks, one with the petrol and one with the pre-mixed insecticide solution. It is injected here. There is a compression. There is a gas, heated gas comes out, then the insecticide, and they are suddenly given release. So they create a fog. These are all the big fogging machines. These are all hand compression, handheld fogging machine, or you can say knapsack sprayer. For there are some modification in this, so you can use as a ULV fogging also. You can use on the truck mounted, on the vehicle mounted. So from here it will come. For the larval applications, you have the hand compression pumps where you use the cone nozzle. You don't use the flat fan nozzle, but for larger area, you can use flat fan nozzle. This is a knapsack sprayer, different. These are hand compression sprayer for insecticidal application. But then there are larvivorous fishes also, which you all must have seen, Gambusia and guppy. Very common, very voracious feeder for the biological control. There are other agents also. This is basically for IRS. If you are using hand compression sprayer, you are using the flat fan nozzle, and there is a recently the standard quality which is pre-qualified by WHO. They have given that you should use the hand compression pump with the control flow valve. The control flow valve between the nozzle and as you are fixing. So what happens? It even if the pressure is reduced, it maintains the swath. The swath is maintained, and you have to use this 8002 E flat fan nozzle. What does it mean? That 80 means 80 degrees swath for liquid output.
Rahul, yeah, thank you.
So 8002, 80 is the angle of the coming of the swath, and then this 02 is 0.2 US gallon, because US gallon is made and the standardization is done. So still they are using gallons in USA. So it is almost equal to 757 ml. So that is why 750 liters it is taken for that particular amount. This is the techniques, but now there are many manufacturers which have revised, and they are coming out with good. This is a polyvinyl. This is a fiber type of nozzle, but always it is good. But please make it a point that don't put anything inside this nozzle to clear it when it is choking. Then what are the options? You have seen all the tools, different situations. What should be the priority? The priority should be We should have the knowledge, we should have the tools as we have discussed in integrated vector management. But what are the evidence generated? Based on that, we land up in different kind of transmission setting, high, low, moderate, and low, and very low. So what options we can use? Now, if you see for vector control purposes or the monitoring purposes, high to moderate, if you club in one group, you can use density first thing, very, very important, density and intensity of the transmission. Where you can use at sentinel sites for routine entomological surveillance, either monthly, quarterly, or during peak transmission season, depending on your resources or your kitty, if you have been given properly to plan and make a strategic plan. So you know your kitty, so accordingly you can make a plan. You have to have spot check sites in area with a specific problem. If you have done stratification, you can use spot checks. You can fix your spot checks like sites in those especially, and then go for conducting the entomological survey. In low transmission season, in low transmission areas, from high to moderate one, and then second category is low transmission area. Density is always measured. You have sentinel sites because sentinel sites was fixed in high. Now you have entered into lower. So the sentinel sites will remain fixed. You can only change the frequency. If you cannot do on monthly basis, you cannot do on quarterly basis. Minimum, the first priority should go for peak transmission season. Then you can expand pre, during, post. Then you can expand quarterly. Then you can expand monthly. So whatever your resources are allowing, that feasibility you have to see. And in this low transmission area, the foci which is active or the area which is reporting persistent transmission, at least one spot check sites we should fix it there. So these are all establishing sentinel sites for low sentinel. If if you feel that there are even in low endemic area, you are not getting the mosquitoes enough in your sentinel or spot check sites, you can always use additional spot check sites for more careful prioritization, more careful monitoring, and then you can prioritize the area. Okay, if suppose there is one area which is persistent transmission and this is area bordering area is not, so you can fix somewhere in between so that it will be representative for this area. So these are all situation based intelligent monitoring, and that is what is known as entomological intelligence. If specimen are less, that is what the question was. If specimen are less, your human blood index, you are not getting adequate mosquitoes. The sporozoite rate you cannot monitor. Entomological inoculation rate you cannot monitor. So this becomes unreliable. If you have facility for the PCR, you can do it. But if you don't have, then what are the priorities? This priority is different, but mandatory is different. Right? So what priority you are going to fix? You have to go for vector species. You don't get any vector, that itself it gives a proof. So if you are not able to do all these things and you are not getting vector, that itself it is a proof that you don't have a vector mosquitoes for transmission of malaria, and you have already got no case, zero case. Frequency and status of insecticide resistance is another problem. This should continue at least because this is done once in a year. Assessment of relative receptivity. This you can have that because see, the area was completely forested and there are some area which has been cleared off and there is no person which is coming residing in the forest range area, or there is complete guarded forest has been made. There is so much development has taken place that neither anybody can enter nor anybody can go out. So the human being is not there. So there will be no malaria transmission. Another example is that if there were some ponds which has been cleaned and they have been made as a recreational ground or they have been made as a tourist spot and proper health care is taken, proper sanitation and proper larviciding has been taken, so the risk is gone. So relative receptivity has to be assessed. The second one is entomological surveillance in sentinel spot check sites will be covered, but it should be based on epidemiological data and local knowledge of malaria risk. Very, very important. And local knowledge of malaria risk is receptivity and vulnerability. If you have cleared any area in the border side and you see that there are a lot of migrations, you should try to understand where these migratory people are going. where they are actually settling. So that resettlement colonies or settlers area, you have to be very watchful and make this. Whether you do mass survey, whether you do rapid survey, whether you do the total preventive measures you take sanitation drive, you take environmental measure, you take source reduction, whatever you measures, you have got so many tools now in your hand. So whatever best feasible with your resources, you can do it. Now, the third is area with changing transmission patch. That is what I was just explaining, that if population is, movement is there, and they are, for some times, they are coming from imported, they are malaria endemic area, and they are settling at the border side. Or, like one example, there are some laborers which are actually on the contract basis in a group of 200, 300 people. They are brought by one contractor for some construction purposes in different infrastructure development purposes for developing the urban area or township or industry. So where they are going, you have to be very good. And then immediately fix one spot check site so that some regular monitoring is done. And then you will get an idea that whether the Stephen size is developing in that area, or this area is under dam construction, so fluid tail is affected area, or it is in the slow running forest area, so it is for the minimus area. So all those things entomological surveillance will lead. So please do that. Make further going down. If you are for high to moderate and low transmission, you are entering into the very low transmission, or you are achieved almost elimination setting, which we do expect by the end of 27 or 26. We will be reaching almost all the area for all the countries which are listening here or which are trying for elimination, which have targeted. Routine entomological surveillance should not be missed. Number one, information on receptivity should not be missed. This is very, very important, that you have to be very, very watchful, especially on the elimination settings. Where intervention is required, you must pinpoint and write down and then give it. If you don't get any idea, suppose you have fixed one sentinel sites here and suddenly you see in the same area, the other side of the village or other side of the some other nearby villages, the people are coming as a migratory population and they are living for 15 days or 20 days or 1 month. You need, if you have resources, you fix up one immediately additional sites or spot check sites, or you shift that sites, or they are permanently settled there. Like one example I give for the Surat Corporation city in India. This is very rich city for the diamond cutting business. So people, they come from endemic areas of different states and they settle. They have got permanent home there also. They come there and they stay for 6 months. So if 6 months they are staying, they are bringing the cases and they are settling. So there is a chance of imported malaria. There is a chance of reintroduction. There is a chance of establishing the transmission indigenously. So you have to be very, very watchful. So your sentinel sites or spot check sites must be reallocated if it is not done there. So these are certain examples which every time you have to put your intelligentsia and take it ground on that. Very low transmission elimination area. Again, if you are having the low transmission, you are actually expecting, everybody expects that you will narrowing down, but you are likely to get some foci. Suddenly you see out of 50 villages, the area is gone, one village is reporting one case. So a small foci you may get. So instead of getting panicky, you should immediately have that much infrastructure. You rush there, do the entomological surveillance, assess what has been the previous report for that entomological surveillance, fix this, set right one is spot check sites, monitor it, liquidate the foci within 7 days, but keep in observations. So observation period will be a little longer. And that is how you have to classify indigenous, introduced, or and then once you have liquidated, just don't remove that spot check site which you have reallocated or fixed. Monitor it, and then you will be able to say after a few times, or maybe after 1 year or 6 months, no issue. Now we have completely liquidated this foci. This is very, very important. This is, if you see, yesterday I showed you a chart for different parameters for groups. Now today you see high, moderate, low in one category, very low to elimination is another category, and then finally prevention of re-establishment if you have finally. So these three category you are going to face. Which parameter, which indicator is very, very significant in this area? Occurrence means prevalence of the vector, presence of the vectors, all the three area it is very, very important. And all the four interventions are very, very important. That is LLIN, IRS, LSM or habitat modification, source reduction. Density is also important for at least high and low, but when you are entering prevention of re-establishment, you may not get density. So density monitoring trend analysis may not be relevant. But all these four parameters have to be always important. Adult vector behavior, biting rate is important for high, low and very low. But in again prevention, that is the last phase, that is category 0. It may not be relevant. Similarly, LLI and IR. LLI which has been given, that will be continued to use. IRS will always be, should be ready for the foci liquidation. But larviciding and habitat modification, unless the area is receptive, the human biting rate will not be important, indicative for the larviciding and this thing. There is no direct correlation for this. HBI, human blood index, biting time, biting location, these are all for the adult. So see, high to moderate or low, this is equally important. But in very low transmission, human blood index will not be important because you may not get the samples. And in 0, yes, it will not be important. But this LLIN will still continue except for the resting locations. But IRS will be important for HBI or they have got direct for the resting. But LSM is not, source reduction is not directly correlated. Similarly, for the adult vector resistance monitoring, for all the three situations which you are encountering, all the three are important. Especially, it is little less important in the 0 category or the prevention of re-establishment category. But you see, LLI and IRS will continue for both resistance. LSM to some extent, because sometimes we do monitor larval resistance, but habitat manipulation environment has no relevance with the resistance monitoring. Intensity and mechanism, all the two, it is important. But for prevention, you may not get any mosquito, infective mosquitoes or long-lived mosquitoes. So there is no. And that is why this LLI and IRS will definitely continue to have the importance of the resistance monitoring, because they are directly connected with their relevant for this. For larval habitats, occupancy and larval density, you see all the three zones, this larval monitoring, it is very, very relevant. And that's why LLI and IRS parameters for this intervention, they are not directly connected, but for larviciding, biological control or habitat modification, these are all directly related. So this is very, very important for the priority area. Sporozoite rate, you may have relevance of this in high to moderate or the low or very low elimination. Once you declare that you have got 0 transmission and prevention of re-establishment phase is there, you have to be very watchful. So sporozoite rate becomes not that relevant because you may not get. Similarly, EIR will be important because you require a lot of mosquitoes. So high to moderate only will be, but very low and prevention of re-establishment, it is not related. They are all directly related. These indicators are directly impacted by LLI and IRS and all the 4. Receptivity, yes, all the 3, it is important and they are directly connected. The larva is there, congenial atmosphere is there, adult will come out, then it will have IRS, it will have impact, LLI will have impact. So all the 4, it will have the impact. Finally, the flexibility. Whether we should have flexibility in both vector surveillance and vector control. Yes, we should have the flexibility, but there are certain things which are non-negotiable. This we have highlighted in many of the platforms. What is flexible? What is non-negotiable for vector surveillance? If you don't have HR, you cannot do it. You don't have skilled entomologists, you cannot do it. You don't have the capacity induction training at the time of introduction. The moment you hire a person and you start asking them to please do this, do that, You will not be able to do, because you have to give motivation and give the training to bring them at your, for your own, to meet your own requirement. Vector prevalence, breeding source mapping, minimum 10 days field work. Field work means entomological collection, entomological field work does not mean they go for supervision of other things, data collection. IRM once in a year minimum. These are all the parameters which are non-negotiable. The another non-negotiable item is, another flexible items are where you can have little more flexibility. You can take innovations according to situation based. You can take your own decision, and that decision will come when you require LLINs. If you have given some LLIN based on some norms to different sizes of the bed nets, but you require in that particular area, no, this size is more required and this size is not required. So you can have flexibility depending on the user rate. Otherwise, if you are very rigid, you give single bed net size, and there are nobody is using single bed net size, they use double bed net size. So neither they will use single nor they are not supplied with double bed net. So that there the flexibility. Now you work out the norms with the population as per the recent WHO norms 1.8. 1.8, one bed net for 1.8 persons. Earlier it was 2.2. Earlier it was 2.5. So 1 person for one bed net is not there, because there are bigger bed nets also supplied in the program. Now, what flexibility is required? You have some residential school where individual students has to be given. You have got hospitals, you have got slum dwellers where some smaller size bed nets, because bigger size bed nets will not be working. Hard to reach areas, tribal pockets where sometimes you are not able to assess properly because they are divided into different hamlets, different pockets. So many of the places you see only one house in one pocket. And if that house is left, you have not made the whole bridge intact. So you have to have flexibility at the ground level, at the local level, that okay, you can divert this, you can change these norms, and you can do. So that flexibility should be allowed. Change of sentinel sites, we have been discussing about that need-based spot check sites or sentinel sites, or reallocation or fixing additional sites. So there should be some provision for that. So these are the places where you can have the flexibility for doing these tools and technology. Similarly, for vector control, there should be complete skill on making the total solution. There should be complete skill for what should be the spray, how the quality spray, how the doses are applied, how the dose is received. Though in program there are certain guidelines given, but there are certain norms which has been given also. So people across the ground level or field level, they should follow that norms. Now, the one thing should be there in their mind, that every time if they are not using quality spray, they are not using proper insecticide, they are not using it judiciously, it is basically they are actually facilitating the precipitation of resistance. So what is non-negotiable in this kind of vector control operations? Quality of tools. You should not use the leaking pumps. You should not use the faulty devices. You should not use the faulty devices for larvicidal application, for any fogging operations or anything. You should check it before that. There should not be any adjustment in that. Dose application, they should be do it properly. If they require that the dose is not proper, they should work out at the local level. They should come out, they should propose that we require higher dose, but resistance data and their inefficacy should be produced. Then the commodities which are approved by the regulatory authority in the country, they should always give the honor to law of the land. There is a system of changing the commodity, introducing new commodity. They should not, nobody should advocate that this is WHO recommended, or this is manufacturing unit has recommended, this is available, this is approved by these regulatory authorities, but this is not included in the program. So forget about the program, use it. No, never. Please don't do that. You should have the regulatory authority approval plus the program recommendation. This is very, very important. Then the capacity building, not only for the in charges, implementers, real operators of the machinery, they should also be given training and they should be given proper time so that they are not lethargy, they are not feeling tired, they are not feeling fatigued. And then legislative measures should be very, very stringent. People, they must know that if it is this is their responsibility, they will be held responsible, they will be accountable for this. So there are punitive measures. As you do the driving, you do wrong driving, you are penalized. You don't have the license of the driving, you cannot drive. Similarly, you don't have a license to create a breeding of mosquitoes. So if you are creating a mosquito breeding repeatedly, so after one notice you are not rectifying, you are liable for penalty. So these are all the flexible things, as I have told you for LLIN also. So this is all what about I just wanted to tell about the tools and technology and innovations. I've tried to answer some of the questions. Thank you, Dr. Leo. Please, over to you.
Well, once again, very comprehensive, thorough, detailed, clear explanation of the issues and how to go about applying or thinking and approaching these issues. So, Dr. Pradeep, we are all grateful to you for that masterclass. Very well done. I'm sure our audience is very grateful for that. So we have a number of questions. I wonder, Dr. Pradeep, is it easiest we just start at the top and you know take them as they come from the top of the list? Do you want to do that on your screen? Should I read it?
Sorry, I have just opened Q&A box. Some
Okay.
of the questions I tried to answer during this your break time, but still I'm very happy that there are many questions. If please
Good.
excuse me if some of the questions I'm not able to answer.
No, of
I will
course.
also try to learn something from all of them.
Yeah. All right.
So
Let's just deal with them. I'm not sure we are going to be able to get to all of them,
so
but let's see how many we can do.
So if
Thank
I
you.
start from top, anonymous, what is anonymous participant? During outbreak or sudden increase in malaria cases, is it still suggested to do human landing catch to determine transmission or not necessary? This is congruent to the assessment of the vector control being used in the area. When there is an outbreak situation, whosoever has asked or for all the information, when there is an outbreak situation, we don't go for the entomological parameters which are done in routine measures. We try to assess what is the purpose of this landing collections or biting collections. They should be done in routine measures and the preliminary survey so that we know that which are the vector prevalent in that area. If we have not done, an outbreak has occurred, first thing is that make a containment zone, try to use the resources available and interrupt the transmission. And then if you are able to have a very strong entomological team, first thing will be that density, abundance, then your total collection, total catch collection. Then you can always use beyond the containment zone, you can use that what is the neighboring area, what is that area. If you have still have lot of resources available, then yes, you can fix up landing collection that whatever in first 3 days or 7 days you are taking the measures to liquidate or check the containment area. If you have adequate number, you can do it. But because you will be doing lot of measures implementation, you will be doing to contain the outbreak, I'm afraid that you may not get the landing collections immediately. We have tackled lot of outbreak containment, but because the first response is that immediately with the insecticides you go and attack. So you may not get adequate number of mosquitoes in that area. What you may get in cattle sheds more number because there will be a lot of diversion of the mosquitoes from the human to the animal shelters. Then another question is that calculation can be given for all frequency Basically, the whole thing was that I did not want to take 1 hour class for insecticide resistance. So frequency, I have told you one calculation I have given the total number of the percentage positive after the 1 hour exposure time. So you can go on doing, because insecticide resistance itself, it is a class of 4 or 5 days. That is mathematics of spray operations and insecticidal resistance. But I have given one small formula. If Muthuras you have not seen, please look into those, and I think that will be hosted on the Orient website. Then there was one question which was answered for Dilruba. Dr. Srivastava, please let me know that which aspirator mouth mechanical is recommended by WHO for HLC model, human landing collection mode or company. See, look here, we are old people. We have been using hand operator and this aspirators or hand collection by the suction tube. But nowadays it has come. See, initially when it came, the mechanical aspirator, it was creating lot of sound. You have to be very, very skilled when you are bringing the mechanical aspirator near the mosquito, because they're very sensitive, very sensitive. So first thing you must stress it that it is good when you are doing resting collection, but when you are using landing collection, if you are very expert, yes, you can do it, because this is basically a technique and technical expertise. But otherwise, when you are doing mosquito sucking, the mouth suction aspirators, we have felt it that is more easy, convenient. It all depends on your expertise and your skill development. Then there is a what is the benefit of the classification of resistance as high, moderate, or low? My question is that if 5 times resistance is sensible, then the current insecticide use is still acceptable. No, there is. I have tried to explain it again. I'll try to explain it that with the tube test, if you have got 98% mortality, means the mosquitoes are susceptible. They are being killed. If you have got, see, 90%, less than 90%, mosquitoes are surviving, it means there is a confirmed resistance. But you are getting. So if less than 90% mosquitoes, I mean, survival is more, and you are getting epidemiological impact, your cases are going down. It means your preventive measure may not be that effective to bring down the dent so fast. And the resistance is one reason for that. So that is why if cases will be reducing, so you will not be able to monitor the trend. You do not know, you are not 100% sure that entire cases you are capturing under notification, reporting. You are missing private, you are missing other dispensary, you are missing corporate response. So what is happening that you are far from the actual number of cases. That is why this insecticide, which is say for example 4%, you are getting less than 90% mortality. So now you have increased this dose. So if mosquitoes are being killed with the 5 times more, it is susceptible or private. But if it is still surviving with 5 times more, then this resistance level goes up. If 10 times more, again it is resistance, you have increased the dose, still mosquitoes are surviving, it means they are 100% resistance. So then there is no option, you have to change the insecticide without considering epidemiology. Then thank you so much for the lucid itna lecture, WHO tube test bottle. Sir, did you use the chemical control in hilly stream areas? How do your country operates? Then it is chemical control in hilly areas. You see, hilly areas, as long as minimus, if you are talking slow running, moving stream, for adult control, you are using chemicals as an indoor residual spray, or you are protecting your community through use of LLIN. User rate should be very high. That is for the chemical use of insecticide treated nets or the indoor residual spray. But if you want to use larviciding application, in case of urban areas, there are many methods, but one method I'll highlight here. There is a bucket and boom method. So what you do, you make a boom, oil boom, and just touch on the top of this where from slow running streams are coming up. If you want to get rid of the larvae, but this is not more recommended in the rural areas because of many such kind of breeding sites will be there. But sometimes it is used and it has given a dividend because oil booms, they will just make a monomolecular pair. They will come downstream and all the larvae will be killed. Flushing is another variety. But yes, chemical for adults, it is being used. Then what is if one insecticide used in an area continuously for years, I want to know in how many years will they develop resistance. Usually when the laboratory it has been done, resistance monitoring, you see from F1 generation, F2, F3, F4. So if you are continuously giving the exposure, after 20 generation, you see the resistance starts precipitating. That was very long back. There was a paper publication in that. But because nowadays you are not isolating a strain, susceptible strain, and then trying to study. So you are in the field, so you don't take that chance. If the area is continuously, there is a separate topic on insecticide resistance management, either by rotation, by mosaic, by there are so many other methods. Then there is during the survey period, sometimes we found places with no larvae pupae, but the habitat is super perfect for breeding. What can be the reason? You have to search more and more. That's what I told you. If you are there is a cases and if you are not getting, breeding sites are there. If you are not getting, either your survey is incomplete or definitely mosquito has changed their behavior. You are not able to reach. There are examples like that you go in the core of the jungle or microphoci, you will see the larvae inside the breeding. How the larval density, number of larvae per deep can correlate with the malaria transmission dynamics. No, it is not directly connected with the malaria transmission dynamics, but number of larvae per deep is measured for high, moderate, low transmission area, because you have to see the receptivity also. Because the moment you are entering into the another phase, low phase, you should see that area is breeding. So anytime any havoc can come. What entomological tests to apply to monitor behavioral changes in Anopheles related to the warming in the indoor and outdoor temperature. This is called as ecological succession. You have to study continuously on monthly basis or quarterly basis as a long-term study for the behavioral pattern density and their abundance and their resting sites, so that you see indoor, outdoor, where the area which was not reported for one species, now suddenly they are reporting. So on the temperature side, and there is another climatic effect that the more the temperature slowly goes up, usually it is says that 2 degrees. So if you go on the Himalayan region, you see the area which was cooler, there was no breeding. Now this is slightly hotter, so you are seeing the breeding. Now once the breeding is established, there is a risk for malaria transmission. And Usha, is it not desirable to do vector mapping in elimination settings so that the output of such an exercise can be compared with the receptivity and vulnerability of an area for guiding the control measures and to sustain the already? By all means, you are right, Usha. This is 100% required, but it all depends on the resources. The vector mapping which is done previous, all the parameters which are done previously, it has to be compared after your impact. That is what is impact evaluation. So monitoring and evaluation, this makes a part that vector mapping, one vector, and vector mapping will come with the vector abundance. That is why if you see the vector abundance has been given in all the three intensity situations: high to moderate, low, and very low, and even POR. Very right. How many specimen count must be there for right HBI sporozoite current minimum? There is no target number like this setup. Whatever you get, please try to do that. Basic mathematical problem is that you should never multiply. You should always try. If it is more than 100, division is always better. But if you are not getting, you can do. There are areas, malaria cases for last 2 or 3 areas. Suddenly, one malaria case is reported from the pre-elimination area. The patient was no evidence to travel malaria. What is your observation? Definitely, there is a. You see, yesterday I told you, if there is, I was giving the classification, case classification, and then FSI qualification, and then unclassified. Unclassified means if you are not able to do, they don't have the case history, but it is a cleared cut FSI, but then you don't have any other thing, you have to label it either as an indigenous or introduced. You cannot call it as an imported. How can countries collaborate the vector control effort, especially in that is what the cross-border collaboration meeting, every time it is being emphasized. I am seeing for the last 20, 30 years, I am seeing these kind of meetings being done. But you know, the regulatory bodies, the decision-making bodies from this side and that side are different. It all depends on the logistics supply. Ideally, yes. Ideally, yes, it should be synchronized in terms of time and space and the quantity. You are using synthetic quiet fluid, they should also use synthetic quiet fluid. You are using indoor residual spray, they should use indoor residual. You are using LLIN, they should use LLIN. But it all depends on the logistics and parameters. That is where the gap is, and this gap is to be minimized. Shanti. Your 3 days presentation were very elaborate and informative, like a slide on options and priority, vector surveillance. Can you project a slide on options and priority on both entomological and epidemiological parameters, field-based in elimination point of view? Priority is always for field only, Shanti. Field priority is always on the field-based implementation. It is for the vector control program implementers and policy makers. This whole exercise is to how to make the strategic planning. So nothing is for the lab-based or inside the rooms. So all the priority which I have shown you, out of the tools available, there are options. And out of the options, which you can prioritize within your resources. These are all for the field only. So whatever 3 slides I have shown you, they are all for the field purposes. No. Last question, Dilruba. Hand catch and trap collection. Hand collection is always a very skilled job, and there are variations, relative density. That is why we call it as a term, that relative density hand collection is depends on the skill of the insect collector or the entomologist. I can collect 20, you can collect 50 mosquitoes. Somebody can collect, the moment you go collecting, it will fly out. So it all depends on the skill. But trap collection is fixed. So, and especially in low density area, when you are not getting more number of mosquitoes, you are not able to reach in time. Now we say by and large 6 to 10 a.m. there should be collection. But nowadays with the climate changing, 6 o'clock there is always a full rise full sunrise. So you should go at 4 o'clock, 3 o'clock. You don't have that much resources to get the vehicle and go at 3 o'clock and reach to the village and disturb them, because they are also sleeping. So in certain situations, so it depends on the trap collection. So there is no controversy in it. I think I have finished all the questions almost, Leo. I've gone down. Yes, now there is no question.
Dr. Pradeep, sorry, I'm just picking up. There's at least one here, which
Please.
was for hourly collection. If we continue collecting for 6 to 7 p.m., then take 15 minutes break, and then again going back collection. Is that okay? Yes, that's absolutely acceptable. In fact, it's routine. If you do your collections hourly, you routinely collect for 45 minutes, and then you spend 15 minutes taking a body break, collecting data, recording, looking after the cups that you've put your hourly collection in. So it's fine to collect for 45 minutes and break for 15 minutes. That's standard. Just don't collect for 15 minutes and take a break for 45 minutes. So that's what Dr. Pradeep.
Absolutely right. Absolutely
yeah.
right.
Dr. Pradeep, you discussed this yesterday, but there's somebody who asked in the chat box, I think, is it acceptable to use the same insecticide that is used in impregnated in an ITN, to use that for IRS, presumably in the same dwelling. So you've already discussed that yesterday, but just maybe recap very briefly.
Okay. WHO very categorically says for insecticide resistance management purposes that it should not be recommended to use the same insecticide, same class of insecticide for IRS as well as for the long-lasting insecticidal nets or insecticide treated nets. Now, the challenge for the program is all those impregnated nets, long-lasting insecticidal nets which are available, they are only pyrethroid impregnated. Now only PBO impregnated are coming, but all the countries, they are not having then their own trials and they are not recommended in the program. So by and large, pyrethroid impregnated nets are available. In class of synthetic pyrethroid, there are 4 or 5 compounds which are recommended for IRS also. Delta methane, permethrin, alpha cypermethrin, lambda cyhalothrin, cyflothrin. There are 4 or 5 classes insecticide which are available. They have got certain structural difference in their chemical composition. Now, if you say that synthetic pyrethroid class we will not use for IRS in that area, and you are targeting malaria elimination. So what compound you will be using IRS? You have got only 3 options. You were having 3 options. One was chlorinated hydrocarbon, only represented by DDT for IRS. You had only organophosphorus. You had only malathion as an option. Because phenothion already banned long back, replaced because of resistance development. Then in synthetic pyrethroid class, you have got 3, 4 classes. Then the 4th class, which is there in carbamate group, only 1 compound, bendiocarb. Then there are some compounds which are coming still, sinusoid or niconiconides. They are not yet introduced under the program. So out of this leftover, DDT is gone. Only organophosphate, malathion and synthetic pyrethroid class. You don't have any option. The only option you have to prioritize the area that where you can use indoor residual spray, where you can use, and the insecticide treated nets, it was once distributed, it is there, it will be there for 3 years. It will be there for 3 years because 3 years is taken as an average shelf life. And the another WHO recommendation is that the area which are entering to sustain that gains, you should have definitely sustain the use of LLIN. So if suppose you have given the LLIN say in 2022 and 25, their expiry will be there. So 25 to 28, another cycle of LLIN has to be given because you are targeting to achieve 27 or sustain the gains. So there has to be some duplication. Another important challenge with the selection of the insecticide is that insecticide shelf life for IRS is for 2 years. The WHO insecticide resistance management plan says that yearly you rotate as a mosaic virus or as a change of the insecticide. The insecticide, when it is manufactured, it is supplied to, by the time it reaches, there are 6 or 7 months or 8 months are passed. It has got 1.5 year shelf life remaining. You cannot waste that insecticide which we have purchased. For a small country, for a small population, it may be good that you can purchase a very small quantity, you finish off and then you can go for next group. But for a country with a population, say example India, when it takes from 200 kilometers to 2000, 3000 kilometer transportation cycle, it will be very, very difficult for them to meet these kind of answers. The another important point, the last point which I will say, when logistic management, when you are advertising for purchase insecticide, if you put that only one insecticide you want this year, say for example, I want delta methrin 500 kg. Then next year I'll say I want lambda cyhalothrin. Same class we are rotating. But then this becomes a brand name. This is never allowed in any procurement procedure. You have to give a generalized tender of the specification. So synthetic pyrethroid in general, and you work out that your per million population. Tomorrow that calculation will come. That For 1 million population, you require delta meth in this much quantity, you require lambda 10% this much, 2.5% this much. Then you work out the cost, whichever is cheaper, you purchase that. So by default, you don't have any choice to select one insecticide which is different than the insecticide which is purchased and which is given in the LLIN. Thank you.
Dr. Pradeep, yeah, thank you. So just for our audience, there is light at the end of the tunnel. I know there are some African countries, Sierra Leone, for example, that are now using clothianidin, that neonicotinoid that Dr. Pradeep was referring to. And I see there's also another, that's a completely different class to organophosphates and pyricarbamates, et cetera. And then there's another one very recently approved, Vectron 500, I think it's called. But, you know, you have to look at cost factors as well. Countries are constrained by costs. So, you know, it becomes a compromise to some extent, and all these factors have to be brought into consideration. So there was one question addressed to me directly by Jenny, asking about short malaria vector surveillance courses. Yes, Jenny, APMEN does offer a 2-week intensive face-to-face vector surveillance course, top quality, each year. Last year it was held in India. This year it was held in Indonesia. We're still finalizing arrangements for the course next year. And we invite nominees. We send invitations to the National Malaria Control Program Director to nominate delegates to that course. And we select 25 people to participate in those intensive 2-week vector surveillance courses where we cover all these topics and so on. And we cover all the expenses, but it's a very competitive process, and we rotate. You know, like last year was South Asia. This year it was the Indo-Malay archipelago. So it depends where you are, but we do offer these courses. Dr. Pradeep, again, a wonderful, highly enjoyable, highly instructive session. We are all so grateful. And tomorrow you're getting to some really practical, nitty-gritty issues, dosage calculations, surface coverage, all that kind of very practical issues one has to consider during your planning for, you know, a control program. So we look forward to that. We thank you again for your time. Thank you to our support team, Dr. Pon, Dr. Popo, Wulan. Very grateful to you, to our audience members for your patience, your interest. Thank you so much. See you all tomorrow. Be sure to join.